everyone for joining in today. This title of the talk is going to be Site-Specific Profiles of Feminization in Surface Waters of California. And what this does is we think it indicates multiple causes of estrogenic activities. Hopefully you'll see some of the smoke guns in terms of what we think might be involved in this sort of endpoint here. I'd like to thank a number of different people involved on this. The name blue are sort of current students involved and collaborators, and a number of chemists, as you'll soon see, played a role in this, including folks David Sedlak, Shane Snyder, and David Crane were instrumental in most of the analysis there. And our source for most of this work was through CalFed Bay Delta program, which I guess has just changed their name, and then so the USDA Ag Experiment Station. We'll think of toxicology and environmental chemistry. Most of us have been trained to recognize things like pesticides and animal waste and pathogens and things of this nature in terms of stressors to aquatic ecosystems. However, more recently, there's been a sort of growing list of compounds that have been discovered primarily through advances in analytical chemistry, particularly that in mass spectroscopy. And we think about how many chemicals there are in the environment. You can see that there's a huge array of different things, naturally occurring chemicals to those that are man-made, coming from a number of different sources. I love this. This is actually a slide by Christian Dalton. He uses in a number of his PCP presentations, and that stands for Pharmaceutical Personal Care Products, which again constitute this class of sort of emerging contaminants, which is what the US EPA is now calling these things. The thing about this universe of chemicals is that it's often hard to sort of analyze those on a chemical by chemical basis or by actually targeting specific chemicals. And here's one. If you follow this slide, you can see again this universe of chemicals. Not all chemicals are extractable, so consequently you may lose a fair large chunk just because of the fact that the chemical that you're after is not extractable into a matrix which allows you to measure it. Secondly, you may not even be able to separate that chemical from the other chemicals you have in that mix. And even if you can separate it, you may not even be able to detect it. It might have a chromophore, or it might have a that allows you to use the detection methods that we have available. So at the A, if you get down here at the bottom, you can see that target analytes are a very, very small percentage of those known compounds. A lot of them are what we say known unknowns, or most of them are what we call unknown unknowns, if you're a big Russell fan, which I think he's sort of taken credit for that sort of term. But so a lot of different compounds out there that we don't really know, and we don't necessarily have standards for in order to measure them. So the question is, well, how do you measure compounds that are pharmaceutically active or are unknown, for example, or have some sort of biological activity? Well, we know analytical chemistry has some issues, particularly that of bioavailability, and as I mentioned, the whole unknown issue. Bioavailability is a huge issue because just because you measure a compound in any matrix doesn't necessarily mean that it's going to be taken up by the organism. And just because it's taken up by the organism doesn't necessarily mean it's going to get to a particular molecular target in the tissues and cause toxicity. So exposure does not equal toxicity. It's a very common misconception, often highlighted by the media oftentimes in trying to, I think, scare people a bit, but that's just personal opinion. But again, exposure does not equal toxicity. You have to have an uptake, you have to have distribution to a particular target in order to get toxicity. And just because you measure something doesn't necessarily mean it may have any biological effects. So how do you get around that? Well, one of the ways that you can do that is by actually identifying a biological effect that you're interested in. And one we're going to talk about today is that of feminization or estrogenic activity. If you have an assay that has a specific response that, say, is receptor-driven, and feminization, of course, is driven by this receptor called the estrogen receptor, you can actually manipulate biological systems, particularly maybe a cell line or actually a yeast or a bacteria, and put that receptor into that system so that you actually generate a high throughput screening sort of method. Now, assays are nice because they're quick and usually very inexpensive to run, but they tell you the whole picture. It really is more, I guess, amenable to getting the whole picture, particularly environmentally, is that of in vivo bioassays. In this case, you essentially need the whole animal to sort of tell you what's going on. And we're very fortunate in this regard, particularly with the feminization response, because egg-laying vertebrates such as fish actually allow us to do that. If you see a male animal that's producing a female response, you can see that pretty readily, and we'll get to that here in a minute and how we can use that. So using those bioassays, you can actually determine whether or not you actually have a response that is significant. And what you might be able to do is then use the bioassay to drive you to the chemistry. So in this way, chemistry and biology can be used together to sort of answer a question, and particularly if you're interested in some of these particular emerging concern contaminants. Now, most common in vitro assays is one that's called the yeast estrogen screen. And this particular situation is what I sort of just described, is that essentially taking a receptor and you're essentially modifying the genetic material of the yeast so that whenever a compound that binds that receptor is exposed to the yeast, you will actually turn a signal on a lighted response. In this particular case, you get a change in color. This can be measured very easily spectrophotometrically or in a semi-quantitative and quantitative sort of manner. So assuming, and why yeast, for example? Why would you use yeast? Well, yeast are easily grown. You can grow them up very quickly. As long as you have sterile technique, anybody in any laboratory can pretty much do this particular assay. That's why they've been so widely used, and it's very inexpensive. You can actually obtain these particular organisms fairly routinely. Because if you have any estrogen or compound that binds a receptor, you're going to get a biological response. So this allows you to look at mixtures. It allows you to look at a host of different sort of water, if you will, or water extracts that you evaluate. And what you would do is essentially plot the response you're after versus whatever matrix you have. So in this particular slide, you can see a dose-response curve using endogenous binding agent for that receptor, which is 17-beta-estradiol. So 17-beta-estradiol gives you a classic dose-response curve. And let's you go out and you want to see whether or not a water sample or a tissue sample or a bile sample or any other sort of matrix that you're interested in that feminization response, you can measure that in your yeast, use a regression analysis to go over to the dose-response curve and actually get an evaluation of estrogenicity in terms of equivalence to that ligand. This is used quite really already in sort of what we call toxicity equivalent evaluations for dioxin-like compounds. Consequently, it's an additive model where you measure a host of different dioxin-like compounds and PCB congeners that bind what's this particular case called the aryl hydrocarbon receptor. You can actually get a TEQ, or a toxicity equivalent value. The same thing being done here, except you're getting an estradiol equivalency value. So an EEQ is what we would then call that, rather than a TEQ. You can do that in vitro, and what you can do, and this is some work that we just had this accepted for publication. I had a student actually go through the literature in 2005 and actually evaluate studies where in vitro EEQs were calculated using, again, this bioassay, and compared that with the analytical chemistry for specific estrogen receptor ligands, or chemicals that bind the estrogen receptor. And what you see is a fairly strong correlation here between the bioassay and the chemistry. Interesting about this relationship is that when you do see failure, it seems to be where you have high biological activity, but haven't, again, evaluated chemistry properly. Consequently, I mean, this would be probably due to the fact of unknowns or other compounds that are in that matrix that haven't targeted in your analytical chemistry. So again, this is a pretty cheap and inexpensive tool that, let's say if you were a agency and you wanted to determine whether or not I need to actually go after steroid hormones, you use this bioassay or a host of them that do give you the same, essentially the same EEQ value, and use that as a screening tool to go back and then say, aha, I do have maybe 10 nanograms per liter of estrogenic activity. Then I could go back and maybe look specifically for compounds that we would think would bind the estrogen receptor, and those would be things like steroid hormones or other compounds of nature. So it's a pretty powerful tool that I think will eventually be implemented in sort of a regulatory framework, maybe in the next, I'd say, at least 3 to 5 years. So the in vitro response. The nice thing about targeting feminization is that, as I mentioned earlier, there's a very nice in vivo method to do this as well, and that's because fish, not all fish, but a lot of fish actually make eggs, and they're oviparous in terms of their reproductive strategy. So what that means is they have to make egg yolk for those eggs to essentially hatch and reproduce. Egg yolk is actually under control of the estrogen receptor in the animal. So consequently, any chemical, again, that binds the estrogen receptor, and this takes place in the liver, if it binds the estrogen receptor in the liver, you actually get of this egg yolk protein precursor called vitellogenin. Now, because it is made in the liver and it is under what we call transcriptional regulation, you can actually measure the messenger RNA for that protein even before the protein is released out into the plasma and then transported to the ovary where it's incorporated into the egg yolk. So it gives you a really nice sort of tool to measure not only the protein itself, which we can do through ELISA and blotting, but also we can measure the transcript itself, or the mRNA, which is a much more sensitive measure you can use molecular tools to get at. The nice thing about this particular system is it also measures anything that changes endogenous estradiol, which is what E2 is here. So let's say you have a chemical that maybe doesn't bind the estrogen receptor, but maybe changes something in the central nervous system that produces more estradiol, or it diminishes the androgenic response, meaning it makes the male less male-like, consequently increasing more of endogenous estradiol. So what it does is it gives you all of the mechanisms of feminization because you actually are measuring feminization in the animal. It operates multiple mechanisms of action as well as just those chemicals that bind the estrogen receptor alone. So the in vitro system gives you a pretty good handle on what binds the receptor itself down here, but in vivo response gives you the whole complete picture because it also incorporates things that change endogenous hormone levels too. Okay, so at that point, does it have any questions, I guess, with regard to sort of the methods that we're going to be using in this particular response? Okay, I'm not seeing any chat questions, so we'll keep going on. We'll ask again if you have any other further questions on some of this stuff. What I'm going to be talking about is some work that we'll be doing up in the San Francisco Bay Delta, which of course has been brought into, well, even I guess national prominence because of this issue called the pelagic organism decline. This is, again, the little animal right here is significantly altering water distribution, particularly down in Southern California. So it's really important to try to understand what is going on in terms of why this and these other three species, as well as a host of salmonid species, are crashing. This crash seems to have started, although you could argue it's been going on for some time, but the real significant crash has started right around 2000. That seems to be this point of inflection that Ted Summers and his colleagues have been arguing about. And this is to be also taking place in some of these other species. So this is how we sort of came onto the scene in terms of this particular sampling location. And this is some work that that I've been collaborating with David Sedlak on for, I guess, about 3 years now. And what we were doing in this study was primarily targeting salmonids, which is pictured down here, and targeting streams that would be sort of influenced by cattle, by range cattle, or by CAFOs, if we'd ever find any of those. So sites that we chose were not necessarily geared towards the pod, but as you'll soon see, it actually did have some pod implications, particularly this site 8 and site 14. But most of these sites, again, were sort of upstream sort of areas looking for sort of issues related to salmonids per se. And what we did is we essentially went and took water from these locations, and then we extracted that water across these C18 discs. So this allowed the disc to be essentially frozen and sent to our laboratory, where it could then be diluted with a solvent and then evaluated chemically for hormones, which is what Sedlak's group did, and biological activity, which is what our group did. Biological activity that we did use, again, we used an in vitro system and an in vivo system. The in vitro system, rather than using the YES assay, we'd actually use the hepatocytes of rainbow trout or salmonid species because we felt that was more appropriate for that particular system. What we can do is actually isolate liver cells from the fish and do exactly what we had talked about earlier, treat the extract with media, and then look for vitellogenin. In this particular case, we were actually looking at the mRNA. The detection limits on this assay are quite low, less than a part per trillion, so it's much lower than what anything you would see from an analytical chemistry perspective. And then we would partner that with in vivo exposures where we were actually taking trout, again. In this particular case, we were actually injecting them with the extract. And we were doing this over 7 days. And you may ask, well, why were you injecting it, and why did you wait 7 days? Because it was our initial hypothesis that the steroid hormones would likely be one of the main causative features in this sort of response. And so using estradiol, again, as our sort of equivalent compound, we were able to show that we could get a nice dose-response curve using 2 injections over 7 days using estradiol as our standard. So that would allow us then to relate any of the extract to estradiol equivalents in those animals. So we'd be measuring protein here in the plasma and messenger RNA from hepatocytes that were exposed separately. And then you could compare the in vitro and in vivo responses to determine what was actually present in those locations. So out of all the locations that we evaluated, the 2 sites kept giving us consistent biological activity. And that was either from the cell exposure, so this is our in vitro assay, or the whole animal exposure. And what you can see is that consistent activity was seen at this site, which is actually the Napa River and the Sacramento River Delta. Interesting given the whole pod. And these were seen throughout the year at different sampling times from 2006 to 2007. The reason why we focused on these 2 is because they consistently give us both in vitro and in vivo responses. We actually had sites that gave us only in vitro, and we actually had sites that only gave us in vivo. But these were the sites where we saw consistently both responses. So the question is, so those were from extracts that we actually took water and filtered over, again, the solid phase extraction procedure. The question came, well, what about the water itself? Well, this is some studies that we just finished probably about a month ago. We actually took water from the Napa River and the Walnut Grove, which is, again, the Sacramento River Delta site, actually transported the water down to our laboratory, exposed live animals, in this case Japanese Medaka, for 7 days and measured vitellogenin. And as you can see, even if fish are exposed to water by itself, we actually are seeing estrogenic activity in that water. We've got some studies ongoing right now with Gary Scher's lab where he's actually caging animals and trying to actually get feral samples to determine whether the animal animals from these locations that live there are actually undergoing feminization as well. So that should be, I would think, by the end of the year. But the message is that the water itself is estrogenic, either through extraction or through a whole water exposures type of route. So the question comes is, well, what the heck is causing this? So we had done some work with Orange County Sanitation, several studies down here, as well as LA County Sanitation District. And what we would call sort of a TIE, or a toxicity evaluation procedure, would actually take a water or sediment extract, if you will, sprayed it across a solid phase extraction matrix. This is what we decided. We've done several different matrices, and it seems OASIS works the best in terms of getting us the best data and with the highest sensitivity. You could basically fractionate that with different percentages of solvents, which would give you a different biological response. So using this sort of biological-based fractionation, you could then see which one of these fractions gave you the highest activity, and then go after the chemistry. So that approach for at least the Napa River and the Sacramento River Delta. So the Napa River sites here. So in this particular case, we actually used methanol instead of ethanol because it gave us a little bit better extraction efficiencies. So again, we used an in vitro and in vivo response, and you can actually see that most of the activity, again, in estradiol equivalents, count in the 80 and 100% fractions of that particular sample that we extracted. What we could then do is take this particular fraction and then pass it through HPLC fractionation, which would then take maybe compounds, or take fractions where you maybe had hundreds of compounds, fract them into fractions where maybe you had tens of compounds, or maybe 2 or 3 compounds. And it'd be these fractions that we actually had biological activity in that we could then actually do some forensic chemistry and see what's present. So you can see, Napa River, most of the activity in this particular fraction in vitro came out in the 80% fraction, whereas most of the in vivo activity came out in the 100% fraction. We decided to go with the 80% just because it gave us a higher degree of EEQ and fractionate it again. But you can still see there's still a significant amount of in vivo activity, particularly in these polar fractions. What's really interesting about this is that if you sort of mark where steroid hormones come out, they usually come out here. And all the activity was in a fraction where there were no steroid hormones. And in fact, if you recall, we actually measured these samples for steroid hormones and didn't see any. Well, I shouldn't say that. We saw very few detections. And those detections were basically less than 1 nanogram per liter. So we're seeing nanogram per liter values, again, around 50 to 60, sometimes 80 nanograms per liter in the samples. Typically, we're seeing 1 nanogram or less than 1 nanogram per liter in terms of the line. So what this tells us is there's a lot of unknown compounds in there that are causing this feminization response. The same thing is true for the Sacramento River Delta. In this particular case, we did see activity at the 60%, which is where steroid hormones come out, and we actually saw further activity after HPLC in fractions. Again, steroid hormones should be coming out right here at about the 8 or 9 fraction mark. But once we're evaluated for steroid hormones, as well as a host of different compounds, so this is, again, where our collaborations with the chemists come into play, including a number of phytoestrogens, a number of PBCPs, a number of pesticides. I think we had 54 different pesticides in there, steroid hormones, and these surfactants. The only thing that we ever saw in any of these active fractions were the compounds. So again, here's looking at the delta fraction as well as our Napa River fractions. And what you can see is that in the Napa River fractions, we're actually seeing some diuron is in basically all of them. And of them, we actually saw some of these triazine herbicide analytes that were present. We also saw some alkylphenol and alkylphenol ethoxylates, which I'll show you here in a minute, that were also present in all 4 of these fractions too. And here's you can actually see the in vitro and in vivo EEQ values. Again, most of the activity in Napa in the nanogram per liter range, which was very high. So the question is, well, what happens when you combine all of these compounds in a mixture actually give us that estrogenic activity that we saw earlier? And what you can see is that even at a 5-fold higher dose, based upon what the ambient dose is, if we look at all of those herbicides and combine them together, we don't see any estrogenic response in our bioassay. So what this tells us is that it's something other than these compounds, even that they're present, even though they're present in these particular matrices. What was interesting, though, is if we took those herbicides and combined them with nanophenol, or these alkyl phenol ethoxylates, and these are compounds, again, normally found in surfactants, a host of different sort of use categories, you could see that in ambient conditions, we really don't see any change, any enhanced estrogenic activity. However, when we actually do about a 5 to 10-fold increase in those alkyl phenol ethoxylates, we actually do see activity. It's actually greater than if we just look at the alkyl phenols themselves. So what this points us to is that there seems to be this either interaction, perhaps, of alkyl phenol ethoxylates with the pesticide that seem to be maybe enhancing activity, at least the in vitro assay with the trout hepaticides. What was really interesting, though, is we were able to demonstrate this also in vivo when we took, in this particular case, our Japanese Medaka, which is, again, a very small sort of laboratory fish. In vivo, when we exposed the fish to those particular compounds, we were also able to demonstrate this greater than additive response. And you can see in a whole animal, you even get a much greater magnitude than you do just with the alkyl phenols themselves, even in the high dose here. This interaction of the surfactants is nothing new. I mean, we had seen this 5 years ago in a paper that we did looking at 2,4-D and triclopyr. This is just showing the 2,4-D data from this particular publication. What's really interesting is if you model response that you should get, again, using estradiol equivalents, you can do this. If you looked at alkyl phenols themselves, we should see what this black bar represents. If we look at 2,4-D by itself, we basically get about 1 nanogram per liter response. So put them together, if this were an additive response, you should see what these 2 bars add up to, which is about 3 nanograms per liter. What you really get, however, when you add them together is a much higher level. So it's a greater than additive response than what you would predict from the 2 compounds themselves. So what this tells us is that somehow these alkyl phenol ethoxylates, particularly in the low dose sort of categories, tend to be enhancing either the translation of these compounds to estrogenic metabolites, or maybe enhancing uptake of the estrogenic chemicals so that we get this biological response. And there's something, I would say this seems to be playing a consistent role, and we keep seeing this in a number of different sites and samples now. So just a little further, this is just showing some further sampling that we've done with Inga Werner, where we did, in fact, in this case, target pod samples. If you recall, the other sites that we did with David Sedlak were much further upstream. These are actually down into the areas where you actually have those 4 pod species residing. And we were able to get, again, samples taken, liter extracts, filtered across these solid extract matrices that were then sent to us for the biological activity. And then what Inga did was then took those same water samples and had them analyzed for, again, a host of different pesticides through David Crane's group at the Rancho Cordova facility. If we samples, again, we still see sites that give us estrogenic activity. In this particular case, these are vivo numbers, again, using Japanese Medaka, nanograms per liter, again, in the 12 to 25 nanograms per liter. When we measure steroid hormones in these, again, less than 1 nanogram per liter. So there's something else in there besides steroid hormones that's causing the response. And again, it's seems to be, in this particular case, I think this is Cache Slough, and this is a site just downstream, actually, in the delta from the Napa River. So again, consistently we keep seeing those responses. If you look at the chemistry in those sites, iron again seems to be present, and this particular sample was pretty interesting. We got a hit with bifenthrin. We saw the phenols and the octophenols and the ethoxylates associated with those. So this was kind of interesting. Again, bifenthrin was interesting because we happen to have another ongoing study with Jan, and one of my students, May Nilos, who just defended last year on this. We were interested in these because we found out that pyrethroids, in addition to being extensively used and use has been widely growing since 2000, which goes back to the sort of the pod inflection point. The really unique feature about these things was that they were chiral, meaning they had multiple carbons. So for those of you that are non-chemists out there, what this means is if you look at a two-dimensional structure of a chemical, it looks like this. But in reality, at these sites where the stars are, you actually have those carbons coming in and out of the plane of the board, if you will. So they're actually not the same chemical. What that means is, and this, if you look at the acute toxicity, they're the same but not the same. What that means is some of these forms actually have much higher toxicity than other forms do. Well, what has this to do with estrogenic activity? Well, we actually published a paper, again, this is in 2007, and this is with bifenthrin, where the S stereoisomer was actually significantly more estrogenic than the 1R. And it was, go back, the 1R is actually the more acutely toxic. In this particular case, the 1S is actually the more estrogenic. Not only was this interesting, the estrogenic activity, but also from a bioaccumulation perspective, the 1S is actually eliminated more rapidly than the 1R. The 1R actually accumulates more in the liver and in the whole animal. So there seems to be not only a stereoselectivity in terms of response, but also an uptake. So this became kind of, and if you recall, bifenthrin was one of the hits that we were getting in those samples. How does bifenthrin cause estrogenic activity? Well, one of the things we hypothesized, was that if you metabolize either ipermethrin or permethrin or any of these sort of pyrethroids, one of the primary metabolites that you get are these phenolic or alcohol-based metabolites. These are highly, highly estrogenic. In fact, studies shown before that they actually do bind the estrogen receptor. Okay. Some of them are even anti-androgenic in vivo, so there is a potential for estrogenic activity in these things. And if we actually take Japanese Medaka in vivo and expose them to the specific stereoisomers for bifenthrin, you actually do see an antioxidant activity for the S enantiomers here. You also see activity in the R forms too, but the S gives us the most activity out of all those stereoisomers. You see that in vivo, and you also see that in vitro with our in vitro assays. Again, the cis and the 1S cis seems to be the most dominant in terms of this particular response. How? Well, do those metabolites active in our bioassay? And the answer is yes. If you actually incorporate these benzyl alcohols and this phenoxybenzyl alcohol, you actually do get estrogenic activity. And really interesting is that this 3-phenoxybenzyl alcohol is likely converted to the 4 metabolite in the cell line that actually gives the higher response. So what this all means is that the metabolites of the bifenthrin, or in this case permethrin, actually are highly estrogenic, and it's likely that the metabolites themselves are playing a role. It's borne out even more so when we actually found an unknown metabolite that was present in these animals. Found using some spectroscopy that this was a hydroxylated derivative of permethrin, So this is actually also likely estrogenic, such that when you have permethrin, you're actually getting metabolism to this compound and to these two compounds, which are then converted to this compound. So what this means is, from a regulatory perspective, you're actually looking for this, and compound's probably not the problem. What the problem is likely this compound and these compounds. We don't have standards for. The only way that we can measure those compounds through a biological system. And this confirms that basically, again, 1S-cis is the most estrogenic, and it actually makes the highest amount of that hydroxylated metabolite. So that hydroxylated metabolite seems to be, if you go after an analyte and you want to regulate something, that's the compound you need to go after, and not the parent compound per se. So it just shows you the complexity of the problem. In vivo, we do see activity with pyrethroids that actually indicate that they are active in terms of estrogenic activity. This 1-cis is the more estrogenic. We actually see biotransformation in trout microsomes, which again lead to these oxidative metabolites, which tend to be the more estrogenic. Okay, the cis, talk about this, but these are actually the cis is actually more susceptible to hydroxylation. Okay, and extensively hydroxylated, so that again leads us to the production of these specific we'd say unregulated compounds that cause estrogenic activity. What relate to our surface water delta response? Well, as you can see, each of those sites carry unique type signatures, estrogenic signatures. That activity is consistently observed not only in the water extract but also in the whole waters themselves. And now we are seeing pesticides and surfactants. We could see anything that demonstrated one particular pesticide or surfactant today. We have some clues to indicate that there may be some interactive effects between some of the surfactants and perhaps some of the pyrethroids, and at least in one of our samples. So the bottom line take-home thing here is that we basically have these unknown metabolites that are present out there that you actually see these greater additive responses in these surfactant sort of interactions with herbicides, but also with the pyrethroids that seem to be playing a role here. So with that, I'm happy to take any questions you might have.
Hello?
Yes.
Hi. I'm wondering, there's been some work done by Cal Poly San Luis Obispo in the Morro Bay area looking at nonylphenol and tumors in gobies.
Yes.
And I'm wondering, I think that what they're trying to do is get nonylphenols listed on the 303D list.
I do.
Do you think that that is the way to attack the problem, or is that maybe being a little bit...
Well, just let me comment on Lars's work for one thing. I'm actually fairly familiar with that. I think it's a little bit premature to suggest they're responsible for tumor formation, primarily because you have other places in the world where you have much higher nonylphenol concentrations than the present in that particular site. So to base it totally on the Morro Bay study, I think would be, I would not do that. Now, does it mean that nonylphenols are a compound that needs to be evaluated? Let me say that they are banned in Europe, and they're banned, and I'm about the ethoxylate, which is where nonylphenol actually are derived from. Right. In my own personal opinion, they're compounds that are what I call catch-22 compounds, meaning that acutely, they're very, very toxic. The ethoxylates themselves are very, very toxic. The argument is though that they're readily broken down to nonylphenols, which are not acutely toxic, but I think are actually showing some, enhanced chronic toxicity. The difficulty lies, however, in the fact that if you look at them in and of themselves, they don't seem to be that, I would say, in terms of overall contributions alone. But what we're seeing, and I think what other people are seeing, is that when they're in mixtures with other compounds, that they do tend to enhance some of these activities. Is that enough to get them on the list? I don't know. I don't know if I can say that or not. But I do know that they do contribute in a mixture setting to the responses that we're seeing. I don't think that they're contributing, however, to at least the tumor formation that's going on in Morro Bay. But I do think that they definitely can cause at least some sort of feminization and some endocrine responses when they're in combinations with some of the other more potent compounds. Is that a question?
Yes, thank you.
I see one chat question to define what POD means. That's pelagic organism decline. That's basically in the upper reaches of the San Francisco Delta where those 4 species tend to reside. That's what a POD area is. Any other questions?
Yeah, I have a question.
Okay.
Kind of follow up on the last one, where you have water bodies that are showing estrogenic impacts in the species in the water body, but what from? Do you have any thoughts on how to, not having a research facility and just needing to identify water quality concerns? Do you have any thoughts on how to break things apart, or how you'd protect the water or start addressing the issues from a water quality protection?
Sure. We just we're actually serving on a panel with SCORP to actually address that, particularly in oceanic systems. And WRF is actually putting together an ambient water panel as well that will come out with a report on sort of how to, basically coming out with suggested lists of compounds that need to be monitored. One that we came out with, at least in our discussions, and this is a little bit premature, so I don't want to jump on this too much, but if you read the report that we just submitted for water use, and this is more human health oriented, not so much ecological, but I think it serves a similar purpose, is that probably one of the best ways to do this is to use a biological response. And I think you're going to start seeing the USEPA and other agencies go to biological responses in terms of screening for compounds, for manufactured compounds, because we can't use the same toxicological methods that we've used in the past on a chemical-by-chemical basis. Having said that, probably the first group of compounds, or I say biological responses that will be utilized will be probably the estrogenic response pathway. It's been sort of highlighted in terms of the endocrine disruption screen testing committee for evaluation. And right now, the agency has just approved testing for 67 compounds to use the estrogenic response, the androgenic response, and the thyroid response as their first sort of screens. So most of the work that's been done using those biological responses have focused on this estrogenic response. And with that earlier slide, you can actually get a pretty good indication of what's there in terms of estrogen receptor ligand binding chemicals using that biological response. So how do you relate that to, say, of a threshold or some sort of that you would use for 303 eliciting or some of the other sort of regulatory procedures? You'd have to essentially that EE value and calibrate it against, I think, some population response, that being altered growth, altered reproduction, or some population response, to use that to say, okay, if we have, let's 10 EEQs based on an in vitro assay, know that the animals in that system are prone to reproductive dysfunction. Those types of studies have not been done at this particular point in time. Most of the work that's been done has been more of a chemical identification procedure. But I think what you're seeing is it's pretty tough to identify those specific chemicals. So what I think is, to make a long story long, I think what you're probably going to have to do is set that
much like you do to a TEQ for dioxin-like compounds, and say, when we have this amount of EEQs, then we're going to set some sort of regulatory threshold on that. And that's what I would predict would happen in the next few years. But it hasn't happened yet because those calibrations have not been carried out, at least as far as I'm aware.
Great, thank you.
Okay, here's another one. Back in an earlier graph showing vitellogenin induction, there was high concentration in Napa River samples, but less than controls in Walnut Grove samples. Can you explain why this is an indication of endocrine disruption? So the Napa River samples that were just taken in February, the samples I presented earlier, those are samples in 2006 and 2007. Those are just grab samples. So what this means is you're basically going out and taking just a liter of water at one point in time in those sites. I think the significant part of that is that even in Napa River, when you consistently go back to Napa River and find estrogenic activity, even 2010, I think there's a pretty good chance that there's something that's consistently coming out the Napa River that is causing some sort of feminization response. You can actually say it's endocrine disruption until you actually put animals in cages there, or you actually sample animals there and determine whether or not you're getting an endocrine response in those animals. What this does is it leads you, so as a regulator, I would be really interested in trying to figure out, one, are animals in that location feminized, and is there some sort of a population response that's going on in that particular location? If those things are true, then I think you could probably start seeing, you might be able to point your way backwards to saying whether or not this particular is actually responsible. Does that answer your question?
Thanks. This is Cam. I was actually just wondering more about the Walnut Grove sample because it lowered the controls. Was it just more variable in your response?
Yeah. So I mean, basically, what you'd say that the Walnut Grove for that particular sample was above control. So again, for that grab sample, we didn't see a response. And honestly, it was, that's not surprising. If you go back to the earlier slide, in fact, I can do that, I guess, huh? You go back way back to this one. Okay? If you go back to this slide, you can see in some cases you actually do get hits, and in some cases you don't, depending on just that grab sample that you got. So the fact that we got a response in Napa, which again continues to be high, is that pretty significant. The fact that you don't see it in one site, just maybe the fact that it just wasn't there that particular time in January. The fact that we, again, can consistently see something just in that 1 liter grab sample, I think is sort of remarkable, at least in my opinion. But if we had seen anything in Napa, it still would have been, well, we just didn't get the right, the right sample at that point. But the fact that we are consistently seeing it in Napa, is sort of interesting to me that there's something continuously present there. Who knows? But I would, if I had a hypothesis, I would again look at maybe these pyrethroid surfactant interactions. We have preliminary data to indicate that actually does occur in the laboratory. I don't want to submit that yet, but use a preliminary evidence at this point.